If you take a single drop of water from a quiet pond and hold it up to the light, it looks entirely empty. But if you push through its surface tension, you enter a crowded, thriving ecosystem. Our eyes simply aren't built to see it. To breach that visual barrier, we have to manipulate light. When light passes through a curved piece of glass, a convex lens, the shape of the glass forces those light rays to bend and converge at a single focal point. This optical trick takes a tiny image and spreads it across our retina, making the invisible appear large. In 1665, an English scientist named Robert Hooke used early lenses to examine a thin slice of dead cork. He documented these repeating, honeycomb-like compartments. Because they reminded him of the sparse rooms where monks lived, he named them cells. A few years later, a Dutch lens maker named Anton van Leeuwenhoek refined this tool. Using a device no larger than a tiny glass sphere mounted on a brass plate, he examined pond water and became the first person to observe living, moving, single-celled organisms. These observations proved that life does not begin at the scale of a leaf or a limb. It starts with the cell, the building block of every organism on Earth. Take a human cheek cell. It has a pliable outer boundary called the cell membrane. This flexibility is crucial for animal cells. It allows our tissues to stretch, our muscles to contract, and our bodies to move freely through our environment. Inside that membrane, the cytoplasm acts as a busy factory floor where chemical reactions take place. At the center sits a dense spherical nucleus, the control room holding the genetic instructions that dictate how the cell grows and functions. Now compare that to a plant cell. Plants cannot run from predators or move to find shelter from the wind. To survive, their cells are encased in an extra thick outer layer called the cell wall. It provides rigid armor and structural support. Because they are rooted in place, plants must manufacture their own food. They do this using green, pill-shaped structures called chloroplasts, microscopic solar panels that trap sunlight and convert it into chemical energy. They also feature a massive internal water tank known as a central vacuole. By storing water and dissolved nutrients, the vacuole maintains internal pressure, keeping the entire plant firm and upright. The architecture of a cell provides the tools for survival. Rigid walls allow plants to stand and harvest light, while flexible membranes enable animals to move and search for energy. While the cells in plants and animals work collectively to build massive bodies, there is an entirely different class of cells that operate as solo agents. These are microorganisms. Microbes are incredibly diverse. They include spiraling bacteria, branching fungi, photosynthesizing algae, and highly mobile protozoa. They have adapted to survive in boiling hot springs, frozen glaciers, and the deepest ocean trenches. They are also highly active in our daily lives. Take yeast, a single-celled fungus. When mixed into dough, yeast consumes the sugars and ferments them, releasing tiny bubbles of carbon dioxide gas. Those trapped gas bubbles are exactly what causes bread to rise and become soft. In agriculture, rhizobium bacteria live inside small nodules on the roots of legume plants like peas and beans. These bacteria pull nitrogen gas straight from the air and convert it into rich, soluble nutrients in the soil, acting as a natural fertilizer. Other soil bacteria act as heavy-duty recyclers. They break down dead plants and animal waste, returning essential nutrients to the earth while releasing flammable methane, which we capture and use as biogas for electricity and fuel. We can even deploy them as environmental mechanics. In the 1970s, scientist Dr. Ananda Mohan Chakrabarty successfully developed specialized bacteria capable of breaking down the toxic compounds in crude oil, offering a biological solution to massive marine oil spills. From the soil to the sea, these independent microorganisms serve as the invisible biochemical workforce that keeps the planet habitable. But there is another type of microscopic entity that operates under completely different rules. Viruses are considered acellular. They lack cytoplasm, organelles, and the basic machinery required to grow, feed, or move on their own. Outside of a host, they are completely inert. To survive, a virus must find a living cell. It docks onto the cell's outer membrane, forces its way inside, and injects its own genetic code. It immediately hijacks the host's internal factory, overriding the cell's original instructions and forcing it to manufacture nothing but copies of the virus. Once the cell is filled to capacity with newly assembled viral particles, they rupture the cell membrane. The host is destroyed, and the new viruses drift outward, ready to dock on to the next healthy cell. Because they lack their own biological equipment, viruses survive by raiding the machinery of others. This cycle of infection and destruction makes them molecular pirates, forcing every living organism to develop new defenses just to survive. If we pull back from that infected cell, past the free living bacteria, and burst out through the surface of that single drop of pond water, the scale of this invisible world becomes clear. In the upper layers of our oceans, trillions of microscopic algae are continuously photosynthesizing sunlight. Together, these single-celled organisms produce more than half of the oxygen in Earth's atmosphere. Our ability to digest food relies on the bacteria in our gut. Our agricultural harvests depend on the nitrogen-fixing microbes in the soil. The breakdown of global waste, the production of our medicines, and the oxygen we breathe are all outsourced to organisms we cannot even see. We tend to measure life by the massive animals and towering forests we can observe with our naked eyes. Yet the largest, most powerful ecological systems on Earth are entirely dependent on the smallest living structures.